Solitary chemosensory cells (SCCs), also called solitary chemoreceptor cells, are isolated elements located in epithelia of the apparatuses of endodermic origin, such as respiratory and digestive apparatuses. In the aquatic vertebrates, SCCs are also present in the skin. In oral cavity, SCCs precedes the development of taste buds. For long time, SCCs were considered to be typical of aquatic vertebrates. Recently, these elements were also demonstrated in mammals. The SCCs share common morphological and biochemical characteristics with the taste cells located in taste buds of the oropharyngeal cavity. In particular, they may express molecules of the chemoreceptorial cascade such as transmembrane taste receptors, the G-protein gustjuicin, PLC-beta-2, IP3R3, TRPM5. Morphologically, the elements of SCCs are polymorphic. Some of them have an apical tuft of rigid microvilli brush cells. Other elements have secretory exocrine granules and others may have endocrine differentiation. Often these elements are innervated. In the upper respiratory system, the SCCs are contacted by the sensory endings of trigeminal nerve fibers (SUBP) and CGRP immunoreactive. The SCCs of the aquatic vertebrates play a role in food search and predator avoidance. The functional role of the SCCs located in internal organs of mammals is unknown also, if several hypotheses have been advanced. In nasal cavity, SCCs operate as sentinels, being involved in detection of irritants and are important in the protection of the airway. See also: diffuse chemosensory system, chemosensory clusters. References: Barbati A., Criscimano C., Bonatti D., Osculati F. Solitary chemosensory cells in the developing chemoreceptorial epithelium of the valate papilla. Journal of Neurocytology 1998, 27–631–35. Sabarbati A., Criscimano C., Bernardi P., Osculati F. Alpha gustjuicin immunoreactive solitary chemosensory cells in the developing chemoreceptorial epithelium of the rat valate papilla. Chem Senses 1999, 24–469–72. Sabarbati A., Osculati F. Solitary chemosensory cells in mammals. Cell Tissue Organs 2003, 175–51–55. Finger T., Botga B., Hansen A., Anderson K. T., Alamohammadi H., Silva W. L. Solitary chemoreceptor cells in the nasal cavity serve as sentinels of respiration. Proc National ACAD Sci USA 2003, 100–8981–86. Gilbertson Tar, Damick S., Margolsky R. F. The Molecular Physiology of Taste Transduction. Kurapin Neurobiol 2000, 10–519–27. Margolsky R. F., Molecular Mechanisms of Bitter and Sweet Taste Transduction. J. Biolchem 2002, 277–1–4. Gulbranson B. D., Finger T. Solitary chemoreceptor cell proliferation in adult nasal epithelium. Journal of Neurocytology 2000, 34–117–22. Hansen A., Witt M., Hummel T. Unconventional neurons in the nasal cavity of humans. Chem Senses 2005, 30, A55. Hansen A. Unconventional sensory cells in the nasal epithelia of rodents and humans. Chem Senses 2006b, 31, E4. Hofer D., Drenkan D. Cytoskeletal markers allowing discrimination between brush and other epithelial cells of the gut, including ectoroendocrine cells. Histochem Cell Biology 1996, 105–405–12. Hofer D. Drenkan D. Identification of the taste cell G protein alpha-gustjuicin in brush cells of the rat pancreatic duct system. Histochem Cell Bio 1998, 110–303–309. Hofer D. Puschel B. Drenkan D. Taste receptor-like cells in the rat gut identified by expression of alpha-gustjuicin. Proc National ACAD Sci USA 1996, 93–6631–34. Merigo F., Bonatti D., Tisano M., Osculati F., Sabarbati A. Alpha-gustjuicin immunoreactivity in the airways. Cell Tissue Res 2005, 319–211–19. Merigo F., Bonatti D., Diccio M., Osculati F., Sabarbati A. Secretory cells of the airway express molecules of the chemoreceptive cascade. Cell Tissue Res 2007, 327–231–247. Sabarbati A., Krishimano C., Bonatti D., Osculati F. Solitary chemosensory cells in the developing chemoreceptorial epithelium of the valate papilla. Journal of Neurocytology 1998, 27–631–35. Sabarbati A., Krishimano C., Bernardi P., Osculati F. Alpha-gustjuicin immunoreactive solitary chemosensory cells in the developing chemoreceptorial epithelium of the rat valate papilla. Chem Senses 1999, 24–469–72. Sabarbati A., Merigo F., Bonatti D., Tisano M., Bernardi P., Krishimano C., Osculati F. Identification and characterization of a specific sensory epithelium in the rat larynx. J. Comp Neurol 2004A, 475–188–201. Sabarbati A., Merigo F., Bonatti D., Tisano M., Bernardi P., Osculati F. Laryngeal chemosensory clusters. Chem Senses 2004b, 29–683–92. Tisano M., Merigo F., Sabarbati A. Evidence of solitary chemosensory cells in a large mammal, the diffuse chemosensory system in Bos Taurus airways. J. Annett 2006, 209 333–7. Zancanaro Caretta C.M., Merigo F., Cavagioni A., Osculati F. Alpha-gustjuicin expression in the vomeronasal organ of the mouse. EURJ Neurosci 1999 11–4473–4475. Whittier M. Solitary chemoreceptor cells. In Chemoreception in Fishes. Hara T.J. ed. Chapman and Hall New York 1992, pp. 103–125. Whittier M., Kotschel K. The chemosensory anterior dorsal fin in rocklings Gydopsaurus and Ciliata, Teleoste, Gadidae, activity, fine structure and innervation. J. Zuhl 1988, 216–339–366. External links. SCC discovery in fishes.